Hello everyone, my name is Vanessa Lovelynna anak and my metric number is 67984. So today I'm going to present you my research proposal, and the title of my research proposal presentation is Glucose 6-phosphate gene cloning and analysis from Empurau fish, and the scientific name is Tor tamborides. The research background of my FYP proposal is Tor tamborides is the highly commercial freshwater species in East Malaysia, and its selling price is almost 400 ringgit per kilogram, located in the Kapit market. However, the population had decreased in nature, causing them to lose their habitat. Glucose 6-phosphate dehydrogenase gene is a cytosolic enzyme which is involved in the mechanism of pentose phosphate pathway. Pentose phosphate pathway is an important metabolic pathway in living organisms, which is located in the cytosol. The problem statement of my research proposal is, although the function of G6PD gene is known in the living organism, but there is still lack information on how carbohydrate metabolism can influence the Tor tamborides. And the hypothesis is gene cloning will be able to create multiple gene clones that could be used to gain more knowledge to improve the growth efficiency of Tor tamborides in aquaculture practices. Next, we will proceed to the objective of my research proposal. The main objective of my research proposal is to isolate the glucose 6-phosphate dehydrogenase gene of Tor tamborides, and also to characterize the glucose 6-phosphate dehydrogenase gene of Tor tamborides. Next, we proceed to the literature review of my research proposal. And the first point of my literature review is the glucose 6-phosphate dehydrogenase gene G6PD. It's the reduction of NADP plus to the NADPH, which convert glucose 6-phosphate phosphogluconate to provide the cell with pentoses. This is cited by This is cited from the Laliotis et al. 2007. NADPH is the main electron donor for the protection of antioxidant, and also for minimize the control of all cells in the biosynthetic reaction. In the slide, we can see the figure 1. Figure 1 is a three-dimensional model of active G6PD dimer, which adapted from MD Capellini 2008. In this G6PD dimer model, it consists of the coenzyme domain, alpha and beta domain, substrate binding site, and also the coenzyme binding site. NADPH also helps cells to combat oxidative stress influenced by a number of oxidant agents. This is cited from MD Capellini 2008. And in the slide we can see that figure 2. Figure 2 is NADP, NAD plus and NADP plus biosynthetic pathway in a prokaryote, which is adapted from the Sebastian KS 2015. The second point of my LR is the pentose phosphate pathway. It's also known as the phosphogluconate pathway. It's an alternative route of the glycolysis to the processing of sugar that create DNA and RNA. This pathway is to generate the ATP, which arrive in a cytosol, which is composed of two steps, oxidative and non-oxidative. The oxidative phase is the essential to maintain the reduction oxidation redox balance by producing the NADPH. Figure 3 is described about the oxidative phase of the pentose phosphate pathway. In this figure, this shows that NADP, two molecule of NADP plus is converted to NADPH by using the enzyme of glucose 6-phosphate dehydrogenase. The third point of my LR is the fish nutrition. Nutrition is very important in the fish diet. This is because it helps to improve the growth rate of the fish and to improve the production in a short time, and also to control the cost of the production. Protein is a very important nutrition for the fish because protein is a dynamic and a high source for the fish. Dietary protein is transformed to the energy glucose rather than being used for the tissue growth, cited from the Ng et al. 2008. For example of the fish nutrition are such as protein and lipid. Protein is a high source of a nutrient needed by the fish. Dietary protein with an EAA profile are similar to the ideal protein. These proteins are required in a decent level and used more effectively for the growth and protein accumulation purposes. Method cited by Metton et al. 1999. And the second fish nutrition is lipid. Fishes require a significant amount of essential fatty acid, EFA. The optimum dietary lipid for Tor tamarides should be less than 5%, and the optimum lipid level for Tor are 10% and 15%. Increased nutrition gross energy will result in no increase in the growth and the feed utilization efficiency, cited from the Ng et al. 2008. My LR is the liver metabolism. Liver is an important organ in the fish. Liver is the for carbohydrate and lipid metabolism. Lipid metabolism is for the synthesis of the structural and the functional lipid. Carbohydrate metabolism is for the basic biochemical process to supply energy. Glucose is for metabolism of carbohydrate to maintain the production of insulin, cited from Ba-Anantel et al. 1991. And the metabolism of liver is influenced by the encourage the gluconeogenic state. The last point of my LR is the gene regulation. Gene regulation is a process used by the cell to increase or decrease a formation of the properties of functional gene. Protein and non-protein coding gene is the product. Quantitative real-time PCR is efficient method to measure the gene expression. Gene expression profile analysis of microarray was widely used for the functional genomic technique in fishing industry, and the evaluation of gene expression using the microarray proved to be extremely useful to identify the target gene, cited for Nielsen et al. 2010. In Figure 3 is described about the major step of gene expression by the PCR adapted from the web of the biologynote.com. The step in the PCR is first denaturing, 2 annealing, and the third is the extension. Next, we will proceed to the material and method parts. The material that will be used in this research is the triazole reagent, proteinase K, agarose powder, TAE buffer, 75% of ethanol, 1.5 ml of microcentrifuge tube, isopropanol, T100 TM thermocycle Biored USA, Easy Pure PCR purification kit Transgen China, loading die, and KB ladle. For the method, there are three main methods in this research, which is isolation and cloning, bioinformatics sequence analyzing, and lastly the multiply sequence alignment MSA. After maintaining the fish, now it's ready to be isolation. Isolation and cloning. And the first step in the isolation and cloning method are total DNA extraction. Total DNA extraction will be conducted using the triazole reagent. A triazole reagent will be added into the tube that contains the mean tissue of the Tor tamarides. The second step is the agarose gel electrophoresis. It's used to detect the presence of RNA isolated from the tissue of T. Tor tamarides. The third step is the primer design. Primer 3 Plus will be used to build the real-time application primer to locate the forward and reverse sequence of the primer. And the fourth step is the gradient PCR. A primer pair of TG6PD gene will be optimized by using T100 TM thermocycle Biored USA to determine the optimum annealing temperature for each set of the primer. And step number 5 is the preparation of LV broth and LV agar. Step number 6 is the preparation for the competent cell. Step 7 is the PCR purification, which are meant to be eliminate the impurities such as the organic compound on the primer. And the step number 8 is the ligation step, which will be set up with the reagent of 2-time rapid ligation buffer, PGEM-TEZ vector, PCR product, T4 DNA ligase, and dioionized water following a standard reaction measure. Step number 9 is a bacterial deformation step by using a heat shock. Step number 10 is the colony PCR. It is used to identify whether the DNA insert has been completely cloned. And lastly, the step is the plasmid mini prep. Is the overnight culture will be centrifuged in the microcentrifuge tube for 3 to 5 minimum and remove the culture media. The bacterial cell pellet will be resuspended at 250 microliter of the cell expansion buffer. The second method is the bioinformatics sequence analysis, which consists of 2 steps, DNA sequencing and DNA analysis. The TTG6PD amino acid sequence will be predicted by using a BLAST basic local alignment search tool program from NCBI database. And the third method is the multiply sequence alignment MSA. Cluster Omega program is used to analyze the multiply sequence alignment MSA for comparison of amino acid and the sequence between the TTG6PD and other species. Upon the completion of this study, it is expected the relation between the hepatic TG6PD and mRNA gene expression in Thor thamnoridase and how it will influence the growth performance in the aquaculture species. Besides that, through the study of G6PD structure, more information can be yielded on the starch utilization and the metabolism, which enhance the aquaculture practice of Empurau in the state of Sarawak. This is my working schedule for the project activity, which consists of proposal writing and presentation, which already started from 2020 last year and started in the month of October. And then we have the activity of genomic DNA extraction, primer design and synthesis, ligation, transformation of E. coli, DNA sequencing analysis, multiply sequence alignment, construction of phylogenetic tree, and lastly will be the thesis writing and the presentation. These are the list of references where I use as my source to do my research proposal. That's all my presentation for my research proposal, and thank you very much for watching.